the insulin receptor is a transmembrane receptor that is activated by insulin, IGF-I, IGF-II and belongs to the large class of tyrosine kinase receptors. Metabolically, the insulin receptor plays a key role in the regulation of glucose homeostasis, a functional process that under degenerate conditions may result in a range of clinical manifestations including diabetes and cancer. Biochemically, the insulin receptor is encoded by a single gene INSR, from which alternate splicing during transcription results in either IRA or IRB isoforms. Downstream post-translational events of either isoform result in the formation of a proteolytically cleaved I+ or - and I² subunit, which upon combination are ultimately capable of homo or hetero-dimerisation to produce the A per ml 320 kDa disulfide-linked transmembrane insulin receptor. Structure: initially, transcription of alternative splice variants derived from the INSR gene are translated to form one of two monomeric isomers. IRA in which exon 11 is excluded, and IRB in which exon 11 is included. Inclusion of exon 11 results in the addition of 12 amino acids upstream of the intrinsic furin proteolytic cleavage site. Upon receptor dimerization, after proteolytic cleavage into the I+ or - and I² chains, the additional 12 amino acids remain present at the C-terminus of the I+ or - chain where they are predicted to influence receptor-ligand interaction. Each isometric monomer is structurally organized into 8 distinct domains consists of a leucine-rich repeat domain, a cysteine-rich region, an additional leucine-rich repeat domain, 3 fibronectin type 3 domains. FNII1, FNII2 and FNII3. Additionally, an insert domain resides within FNII2, containing the I+ or - /I² furin cleavage site, from which proteolysis results in both ID+ or - and ID² domains. Within the I² chain, downstream of the FNII3 domain lies a transmembrane helix and intracellular juxtamembrane region, just upstream of the intracellular tyrosine kinase catalytic domain, responsible for subsequent intracellular signaling pathways. Upon cleavage of the monomer to its respective I+ or - and I² chains, receptor hetero or homo dimerisation is maintained covalently between chains by a single disulfide link and between monomers in the dimer by 2 disulfide links extending from each I+ or - chain. The overall 3D ectodomain structure, possessing 4 ligand binding sites, resembles an inverted var unregistered trademark, with each monomer rotated approximately twofold about an axis running parallel to the inverted V and L2 and FNII1 domains from each monomer forming the inverted V's apex. Ligand binding: the insulin receptor's endogenous ligands include insulin, IGF-I and IGF-E. The binding of ligand to the I+ or - chains of the IR ectodomain induces structural changes within the receptor leading to autophosphorylation of various tyrosine residues within the intracellular TK domain of the I² chain. These changes facilitate the recruitment of specific adapter proteins such as the insulin receptor substrate proteins in addition to SH2B, APS and protein phosphatases, such as PTP1B. Eventually promoting downstream processes involving blood glucose homeostases. Strictly speaking, the relationship between IR and ligand shows complex allostric properties. This was indicated with the use of scat-card plots which identified that the measurement of the ratio of IR-bound ligand to unbound ligand does not follow a linear relationship with respect to changes in the concentration of IR-bound ligand, suggesting that the IR and its respective ligand share a relationship of cooperative binding. Furthermore, the observation that the rate of IR-ligand association is accelerated upon addition of unbound ligand implies that the nature of this cooperation is negative. Said differently, that the initial binding of ligand to the IR inhibits further binding to its second active site, exhibition of allostric inhibition. Although the precise binding mechanism of IR and its ligand has not yet been elucidated structurally, as identified using a systems biology approach, biologically relevant prediction of the IR ligand kinetics has been identified in the context of the currently available IR ectodomain structure. These models state that each IR monomer possesses 2 insulin binding sites. Site 1, which binds to the classical binding surface of insulin, consisting of L1 plus I plus or minus ET domains, and site 2, consisting of loops at the junction of FNII1 and FNII2 predicted to bind to the novel hexamer-face binding site of insulin. As each monomer contributing to the IR ecto domain exhibits 3D mirrored complementarity, N-terminal site 1 of one monomer ultimately faces C-terminal site 2 of the second monomer, where this is also true for each monomer's mirrored complement. Current literature distinguishes the complement binding sites by designating the second monomer's site 1 and site 2 nomenclature as either site 3 and site 4 or as site 1 foot and site 2 feet respectively. As such, these models state that each IR may bind to an insulin molecule via 4 locations, being site 1, 2, or. As each site 1 proximally faces site 2, upon insulin binding to a specific site, cross-linking via ligand between monomers is predicted to occur, or as, monomer 1 site 2, insulin, monomer 2 site. In accordance with current mathematical modeling of IR insulin kinetics, there are 2 important consequences to the events of insulin cross-linking. 1. That by the aforementioned observation of negative cooperation between IR and its ligand, that subsequent binding of ligand to the IR is reduced, and 2. That the physical action of cross-linking brings the ecto domain into such a conformation that is required for intracellular tyrosine phosphorylation events to ensue. Biological significance: tyrosine kinase receptors, including the insulin receptor, mediate their activity by causing the addition of a phosphate group to particular tyrosines on certain proteins within a cell. The substrate proteins that are phosphorylated by the insulin receptor include a protein called IRS1 for insulin receptor substrate 1. IRS1 binding and phosphorylation eventually leads to an increase in the high-affinity glucose transporter molecules on the outer membrane of insulin-responsive tissues, including muscle cells and adipose tissue, and therefore to an increase in the uptake of glucose from blood into these tissues. In other words, the glucose transporter GLUT4 is transported from cellular vesicles to the cell surface, where it then can mediate the transport of glucose into the cell. Pathology: the main activity of activation of the insulin receptor is inducing glucose uptake. For this reason, insulin insensitivity, or a decrease in insulin receptor signaling, leads to diabetes mellitus type 2. The cells are unable to take up glucose, and the result is hyperglycemia, and all the sequelae that result from diabetes. Patients with insulin resistance may display acanthosis nigricans. A few patients with homozygous mutations in the INSR gene have been described, which causes Donahue syndrome or lepreclanism. This autosomal recessive disorder results in a totally non-functional insulin receptor. These patients have low-set, often protuberant, ears, flared nostrils, thickened lips, and severe growth retardation. In most cases, the outlook for these patients is extremely poor, with death occurring within the first year of life. Other mutations of the same gene cause the less severe Abson-Mendenhall syndrome, in which patients have characteristically abnormal teeth, hypertrophic gingiva, and enlargement of the pineal gland. Both diseases present with fluctuations of the glucose level. After a meal the glucose is initially very high, and then falls rapidly to abnormally low levels. Regulation of gene expression: the activated IRS1 acts as a secondary messenger within the cell to stimulate the transcription of insulin-regulated genes. First, the protein GRB2 binds the PT residue of IRS1 in its SH2 domain. GRB2 is then able to bind SOS, which in turn catalyzes the replacement of bound GDP with GTP on RAS, a G protein. This protein then begins a phosphorylation cascade, culminating in the activation of metagen-activated protein kinase, which enters the nucleus and phosphorylates various nuclear transcription factors. Stimulation of glycogen synthesis: glycogen synthesis is also stimulated by the insulin receptor via IRS1. In this case, it is the SH2 domain of PI3 kinase that binds the PTR of IRS1. Now activated, PI3K can convert the membrane lipid phosphatidylinosytol 4,5-bisphosphate to phosphatidylinosytol 3,4,5-triphosphate. This indirectly activates a protein kinase, PKB, via phosphorylation. PKB then phosphorylates several target proteins, including glycogen synthase kinase 3. GSK3 is responsible for phosphorylating glycogen synthase. When GSK3 is phosphorylated, it is deactivated, and prevented from deactivating glycogen synthase. In this roundabout manner, insulin increases glycogen synthesis. Degradation of insulin: once an insulin molecule has docked onto the receptor and affected its action, it may be released back into the extracellular environment or it may be degraded by the cell. Degradation normally involves endocytosis of the insulin receptor complex followed by the action of insulin-degrading enzyme. Most insulin molecules are degraded by liver cells. It has been estimated that a typical insulin molecule is finally degraded about 71 minutes after its initial release into circulation. Interactions: insulin receptor has been shown to interact with ectonucleotide pyrophosphatase phosphodiesterase 1, PTPN11, GRB10, GRB7, PRKCD, IRS1, SH2B1 and 2 Moroccan dirhams L1. References. Further reading. External links: Insulin receptor at the U.S. National Library of Medicine. Medical subject headings.